talk. We paid for the pizza, yes. It all comes from Joel, anyways. So Henry Van Brocklen, who's my partner in crime, the co-director of CIDMI, is coming right down. He had another call right before he's upstairs, so she'll be joining us. Before we introduce our speakers, just want to give you a little bit of a plug. So one thing that we have been doing is trying to support folks getting involved in molecular imaging in the ID space. And so as Joy can attest to, is our first CIDMI seed grant winner. We had some excellent applications. We also had external review. And so this is a process that we're trying to kind of grow on now as we go forward. So as the first, and now we actually are advertising our second RFA, or RFP, request for proposals. And this is for kind of looking to working in collaboration with someone in the Department of Radiology, Nuclear Medicine, and Molecular Imaging with ID space. Happy to talk to Henry, who just came in here, and myself about this if you're interested. And what we'd love to do is connect you with folks in each of the different departments, divisions, et cetera, to kind of come up with ideas as well. We don't have a huge amount, so we can't offer a large number, but up to 20,000 now for certain applications that we can do. So we're really looking forward, especially trainees, people junior, people who have not worked in this field before, but anybody can apply outside of that. So yeah.
Folks in radiology who may be interested in. We're going to try to reach out to some of our colleagues to try and pull together some of these connections.
Yeah. So yeah, so reach out to Henry, myself, or we can get you connected, or if someone expressed interest and you may have some other idea, we're able to kind of act as facilitators as well within that space. And we'll obviously have external reviews as well for this cycle. So, try not to be too biased in our decision making as we go. So, yeah, so help yourself. You can scan QR codes, spread the word. I believe when is our due date, Val? For the April 24th is going to be the due date for these are short applications, 3 to 6 page, 1 page budget, so not extensive. With that, so what I'll do is I will actually introduce Joy Okoro, who is a third year now. Is that right? Wow. BMS student in the Solomon Lab, and has really been doing some excellent work in the TB space, and really the external reviewers really raved about the project, and there was just a clear interest and clear winner in your proposal. So congratulations. And what I thought we would do is, Joy, it sounds like you want to take questions after, is that right? If you have questions to ask as well during the talk or clarify questions, we'll have a microphone to pass around, because that way people online can actually hear, which has been an issue I know unless you're in front of a microphone. So without further ado, I will introduce Joy. Come on up.
Hello, everyone. Thank you so much, Tim, Henry, and Val, for giving this award. I also want to say thank you to my PI, Sarah, for her guidance and direction in developing this project. So today, I'm going to be telling you about how I'm going to use spatial transcriptomics, specifically xenium, to study how CMV infection alters the local lung landscape to promote MTB pathogenesis. So why are we even talking about CMV, a viral infection, in the context of TB? We know that only about 5 to 10% of people who are exposed and infected with MTB progress to active TB disease. And there are many factors that are known to increase this risk of progression to TB. But more recently, there's been reports that suggest that viral infections can increase this risk of progression from MTB infection to active TB disease. So Fatu and Josephine in our lab actually put out a review not too long ago about the impact of viral infections on TB risk. So they came up with this two-hit model of how acute and chronic viral infections precipitate TB risk. Where's my cursor? Okay, so in both chronic and viral infections, the initial infection creates this bump in TB risk. But in the context of acute viral infections, there's a transient period in time where, due to viral load, there's an increase in TB risk. However, in chronic viral infections, you have this bump after the initial infection, but because chronic infections establish a reservoir and are maintained in individuals, this can result in elevated TB risk that's persistent. And this is due to increased inflammation and systemic and tissue type interferon signaling as a result of viral infection. This is not super relevant for this project, but in context, antiviral therapies that can clear viral reservoirs are able to, or potentially able to, restore TB risk to pre-viral infection levels. And this is a nice model for generally how chronic infections could influence TB risk, but I think it's very important to mention that in reality, not all viruses would lead to this impact. For example, so I think so far in the papers I've read, EBV, for example, has not been shown to be associated with increases in TB risk, even though it's a chronic viral infection. Okay, so I'll give you some relevant background about CMV. So CMV is a highly prevalent herpes virus. And as you can see here, in all the countries from which we have data, about 50 to 100% of the population is estimated to be CMV positive. And CMV tends to be asymptomatic in immunocompetent hosts, but can induce severe symptoms in immunocompromised individuals. It's also ideal for studying interactions with other pathogens because it establishes lifelong latency in hosts. CMV is known to infect a wide range of cell types and tissues, and it induces significant innate and adaptive immune changes within the host. Related to TB specifically, we're interested in CMV because there's certain regions where you have high CMV prevalence also overlap with TB endemic regions. And I guess within the host, CMV and MTB are known to infect macrophages and can establish latency in the lungs. Okay, so on to the epidemiological data we have. So there's multiple studies that have revealed that CMV can impact the clinical outcomes of MTB infection. So this first figure is from a large cross-sectional study from the US National Health and Nutrition Examination Survey. And the authors found that CMV seropositivity was independently associated with latent TB infection. So specifically, they saw that 90% of the participants with latent TB were CMV positive, while only 58% of the people without latent TB were CMV positive. And this association that they saw remained even after they corrected for other confounding variables such as birth location, age, I believe, and Hep C seropositivity, suggesting a clear immunological link between CMV-induced immune activation and TB susceptibility. Another study, which enrolled pregnant mothers in South Africa and followed up the babies for up to 9 years of age, found an approximately threefold increase in TB risk in childhood associated with contracting CMV infection within the first year of life. And this is pretty important because it shows how early, I guess, infection with CMV can change outcomes or can change the risk of TB over the childhood span, I guess. Sorry? Yeah.
Sorry, I'm supposed to speak into the microphone. The curves diverge immediately, which suggests that maybe the population's different for other reasons, or not.
That's a possibility. If I remember correctly from the study, they did do other analyses where they excluded confounders, and they found that I guess what they focused on was the adjusted hazard ratio that was still 3.2 between the two groups, irrespective of the confounders. Outside of this, there's also been a recent paper that showed that CMV status was associated with increased bacterial burden and TB disease severity, as well as altered cytokine expression in a South Indian population. So all this epidemiological data is suggesting that there's potential changes that CMV is inducing in the host that could impact TB risk. So kind of going a little more into what those mechanisms could be. So very generally, CMV is known to induce broad remodeling of the host immune system, and that can have direct or indirect impacts on TB immunity. We also know that CMV replicates in the lung, which raises the possibility that CMV might contribute to an immunoregulatory environment that would promote a local loss of MTB control. Something else that CMV is known to do is to drive monocyte differentiation into macrophages, which are the primary targets of MTB infection. But focusing on this figure on the slide, in this paper, the authors showed that MCMV-infected macrophages upregulate immunosuppressive genes, while the bystander monocytes show elevated type 1 interferon signatures. And this is important because type 1 interferon is known to impair TB immunity and increase susceptibility to MTB infection. So this then potentially provides a mechanism for how CMV-induced lung remodeling would increase TB risk in the context of CMV-TB co-infections. Okay. So now I've given you the background and the scientific rationale for this project. So I'm going to tell you what I plan to do experimentally and what questions I'm looking to answer. So as far as we know, we're developing the first mouse model of CMV-TB co-infections. And looking at group 4 here, we plan to inject 2 to 3 week-old black 6 mice with 1,000 PFUs of GFP-labeled Smith strain MCMV. And then 4 weeks later, we're going to follow up with a low-dose aerosol infection of MTB using the Erdmann and Cherry strain. And for our controls, we have naive mice, MTB only or CMV infected only mice. And the goal for this project is to collect the same lung lobe region from animals in each group, fix them, and mount them on slides for xenium processing. I'm going to tell you a little bit more about xenium in a second. Okay. So for my proposal, I had 2 main aims. So the first was to use xenium to map the spatial distribution of CMV-infected macrophages in MTB co-infected lungs using the GFP and M-Cherry reporters on both pathogens to see if areas of high viral load would correspond with MTB burden and more severe lung pathology. And my second aim was to identify regions of high type 1 interferon expression in CMV co-infected lungs and to look for overlap with markers of local immune suppression and MTB replication using xenium. So what exactly is xenium and how does it work? As far as I understand, it's a combined imaging and transcriptomics platform that's offered by 10x Genomics. It allows you to visualize and quantify gene expression and protein exactly where they're found in intact tissues, preserving the spatial data as opposed to single cell sequencing where you have to homogenize your tissues for your analysis. So for this process, you design a panel of DNA probes that are added to the tissue that's fixed on the slides. So the DNA probes, which they call padlock probes, contain 2 regions that hybridize to your target RNA and a 3rd region that encodes a gene-specific barcode. So the 2 ends of these probes would bind to the target RNA and are ligated to generate a circular probe. And then this probe is then amplified using rolling circle amplification. So you're creating multiple copies of your gene-specific barcode for each RNA target. So your xenium slides with the tissue sections are then loaded onto the analyzer. And here they have these fluorescent probes that bind to your amplified probes from the previous step. And then they do multiple rounds where they add the fluorescent probes, they image and then remove the probes in multiple rounds. And this essentially generates an optical signature that's specific for each barcode. And then they convert this into gene identity. And this allows you to identify exactly where your RNA transcripts are on your fixed tissues. And the cool thing about Xenium is that you can combine this with H&E staining to then combine your morphological data with your transcriptomic data, giving you like a, deeper resolution into what's actually happening in your tissue. So this is an example of what a Xenium imaging output looks like. I've heard it's pretty cool to analyze, so I'm excited to see what my images will look like. But for this project, we're planning to use a custom design panel that includes markers that are relevant to my question. So 10x offers a mouse pan tissue panel that was used to generate the image I just showed. So we're planning to use some of the probes that they included in this panel, so the ones for mouse lung and some general cell types. But for the panel we're designing, we also want to add immune-specific markers for like macrophages to identify ISGs, as well as markers of immunosuppression and immune activation. Immune activation. But more importantly for this project, we want to include probes for CMV and MTB specific transcripts. And we have a few ideas for this, but I'm actually very open to suggestions. One of the things we're planning, or one of the, I guess, avenues we're exploring is so there's an established dual RNA-seq analysis pipeline where they identified MTB ribosomal RNA sequences. So one of the ideas we have for this is to design probes that would target these MTB-specific ribosomal RNA sequences, but I'm open to other ideas for targeting MTB-specific genes. We also plan to use a similar approach to identify CMV-specific transcripts. But as I said, the panel is still in development, and we're planning to get some insights from a couple of people that Tim recommended from his lab. But if you have suggestions, I'm very open. So what do I actually want to get out of this project? The main goal is to design and validate a Xenium panel that will allow us to detect MCMV and MTB in these fixed mouse tissues. But I already mentioned what my, I guess, biological questions here are. So I want to understand how CMV is altering the local lung environment to promote MTB pathogenesis. And I would also be looking to see if CMV infection drives local increase in MTB burden, and if these regions overlap with the expression of type 1 interferon in the lungs. Because this grant is $10,000 and Xenium is pretty expensive, I have to be quite realistic of what I'm able to accomplish. So to maximize the budget, we've decided to mount lung sections from multiple mice on the same slide. So as far as I understand, each Xenium run can accommodate up to 2 slides. So we're hoping to be able to fit around, or at least 2 animals from each group, each of our 4 groups in the run. And we also plan to do H&E and potentially immunofluorescence staining on these same slides after the Xenium run, which would then allow us to directly compare our transcriptomic data to the morphological data, also allowing us to save on mouse costs. So yeah, I hope this has given everyone a big picture understanding of my proposal. I want to say thank you again to CIDMI, Tim, Henry, and Val for giving me this award. I also want to thank my amazing PI, Sarah, for her guidance and support, and the entire Solomon lab. I also want to say special thanks to Val, who's the CIDMI program manager, and also Brian in the Henrich lab, who talked me through the Xenium workflow and has offered to allow me to shadow their upcoming Xenium experiments. So, thank you.
Okay, go ahead and take questions.
Oh, okay, yeah.
Just ask for the microphones to people online. That'll
I have a really kind of basic viral TB question. So we're looking at how chronic viral infection can increase your risk of TB infection.
Yes.
Does it have a similar effect on like latent activation? Do you know?
I mean, latent activation of what?
Of TB.
Oh,
or is it just the primary infection that we're looking at?
That's a good question. I think in the context of CMV, because of how prevalent it is, the assumption is that most people would probably have acquired CMV prior to acquiring TB. But I think it's an important question.
I see. Yeah, it'd be hard to disentangle those two questions. Yeah.
It's worth exploring. I think in one of the iterations of a grant we had written for this project, we also had a condition where we would infect with MTB and then CMV to see what would happen. So I can't tell you right now, but
interesting. Thank you.
Another time.
Joy very clearly expanded. I've got a question, partly because I missed it, and my fault. How old are these mice when you're infecting them with the CMV?
So in the way I formulated this, we're planning to do around 2 to 3 weeks of age. And the reason for that is because I guess the strongest piece of data we have now is from the pediatric cohorts of, I guess, yeah, the pediatric cohort they followed. So what we're seeing is that this increase in TB risk is pronounced in childhood. So we thought about using younger mice to sort of recapitulate.
And in terms of MCMV and timing of infection, what's known about that? Like in children, infants can get severe CMV, although this is post-wean, so it's not really infants. But what's known about the mouse model for timing of CMV infection?
I'd have to look a little more specifically to be sure. From what I remember in conversations with Oscar, I think you still see the same like immune activation effects. I don't know if there's like increased mortality if you infect the mice younger compared to adult mice. I'll have to go back to see, but I remember like talking about it. It's something that we want to, I guess, model in neonatal mice because that's what the data says. But we also have plans to like do the experiments in adult mice to see if there'll be a difference. Yeah.
Thanks.
Very interesting project. I had two just general questions because we've also been considering CIDMI for a bit. About how many cells do you get like per section per sample? And then is it known from the data that's out there, like, is it evenly distributed, the infection across that slide, or are there foci of infections, or is that like one of the questions I would be able to answer?
So in terms of how many cells you can get, I think from the 10x websites, I remember seeing, like, it depends on how you configure the tissues on your slide, but I remember seeing that you can have up to 1 million to 2 million cells like accommodated on a single slide. And in terms of the infection, because we're infecting these mice in vivo, I guess the pattern of infection will be determined by the mice, but we have the reporter strain, so that would actually that's one of the questions I'm looking to answer to see how like infection is, I guess, forming, whether in pockets or more broadly across the lung tissue. Yeah.
Time for one more question. We'll have to
Thanks. Just a quick one, Joy. So in terms of, because TB is patchy disease, so
is what?
It's patchy.
Oh yeah.
So in terms of the infection and foci, one of the things you probably need to consider is before placing on the xenium, you're sure that whatever tissue sections you have on the xenium do have infection, because you run the risk that you may run through and find that probably there was nothing. And because the H&E stain it's post-xenium staining, it's unlike Visium. With Visium, you do it pre-running it. So you'll be sure as to when you are going to image and not. And the other thing that probably you may want to consider is they now also have a proteome panel to go with the transcriptome panel that you might, just to increase your sensitivity and pickup rate. And probably in terms of the TB genes, the other one to probably consider would be if you have make sure that you have the, which I did as a mistake probably to avoid, is to make sure that you have something that you can be able to calculate your RS ratio from the probes that you have.
Yep. That's both good contributions. Thank you so much.
Well, Joy, congratulations again on an excellent, excellent talk. So Tim asked if I would come up and introduce the next speaker. I have no notes, so it's a really good thing that all of you know the next speaker very, very well. And so this is Dr. Joel Ernst. He's going to talk to us today about some of his work in M. tuberculosis. So Joel, thank you very much for agreeing to be here and speaking today.
Thanks, and it is kind of amusing to be listed as a guest speaker. I guess that means it really is a CIDMI seminar. So I'll tell you about it. This is very much a project that's a work in progress. It also has multiple stories embedded in it, but I'll try not to go too deeply into it, other than to start with, this is a project that was first conceived in around 2010 or 11. And it's still a project that's a work in progress. And maybe I'll include a little bit about that reason for that. So in order to kind of follow this I'll just want to quickly review sort of the basics of the life cycle of a T cell. So antigen-specific T cells are primed in secondary lymphoid tissues, lymph nodes, spleen, other secondary lymphoid tissues. They proliferate, they differentiate, they leave the secondary lymphoid tissue into the circulation, and in the context of an infection or inflammatory stimulus, tissue localized inflammation, they basically traffic to that non-lymphoid tissue and become activated again, where they then perform their effector functions and ideally eliminate the infection. Now, there's something I won't go into, but obviously many of you know that once a population of effector T cells develops, there's also contraction, and the contraction involves activation and induced cell death, but then there's a population of memory cells that persists. So the first question that really kind of started this was the question whether for optimal immunity, whether T cells need to directly contact and activate MTB infected cells in the lung, or whether in contrast, and I think what a lot of people would have predicted was that it doesn't matter where a T cell gets stimulated or activated in a tissue, because cytokines are low molecular weight proteins, they diffuse like crazy, and should be able to act at a distance. And in fact, there was an example of that published in Leishmania around the time that this part that I'll tell you about was being done. And it showed that a Leishmania-specific T cell could be activated as far as 80 to 100 microns away from a Leishmania-infected macrophage and still control that Leishmania-infected cell and eliminate the Leishmania. So we wondered whether that model applied to MTB, or whether in MTB the scene was different and direct cognate interaction was necessary. So this is a complicated slide, but it serves as the basis for a lot of what I'll tell you about. So I'll kind of walk you through it. So the question really was, as I just stated, this direct cognate interaction with an infected cell required for optimal control of intercellular infection with MTB. So what we did is we made mixed bone marrow chimeric mice, so irradiated wild type mice, reconstituted their bone marrow with a 50-50 mixture of bone marrow from wild type mouse or bone marrow and bone marrow from a MHC class 2 knockout mouse. So what that generated then, when they were reconstituted, was a mouse that 50% of their macrophages would be derived from the class 2 wild type, 50% from the class 2 knockout. we then infected those mice. And the idea was we wanted to be able to do this with cells that came out of the same tissue from the same mouse, rather than just infect different mice. So we infected them with fluorescent protein expressing MTB, the H37RV strain. Then, as if this weren't complicated enough, and for reasons that I'll tell you in a minute, half of each set of mice got CD4 T cell depleting antibody. Then that was followed by euthanasia several days later. Flow sorting of the infected cells, so GFP expressing bacteria used as the reporter for infection. And then we quantitated the number of bacteria per infected class 2 wild type or class 2 knockout cell. And so what this shows, and two different cell subsets basically showed similar results. But I'll just focus on this one, which is actually I'll focus on this one, which is a subset that we termed at the time recruited macrophages. They're monocyte derived. And what it showed was that the most common MTB infected class 2 wild type cell had 1 bacterium per infected cell, whereas the class 2 knockout cells from the same tissue had 2 to 4 and sometimes more mycobacteria per infected cell. So that indicated that a class 2 knockout cell that could not be recognized by a CD4 T cell was more sort of a better environment for Mycobacterium tuberculosis than a wild type cell. But since various other activities have been attributed to MHC class 2, we wanted to know whether that interpretation was absolutely correct. So when we depleted the CD4 T cells, the difference between class 2 knockout and class 2 wild type cells with respect to bacterial load was basically made to be the same. So the message here then was direct contact, direct recognition of an infected cell by a class 2 or by a CD4 T cell was necessary for optimal infection or optimal immune control. But now here's the rub. In tissue lesions, granulomas, T cells and infected macrophages are found in 2 different sections or regions of a granuloma. And so this is in 3 different species. Human, for complicated reasons, the color scheme is a little bit different for each species here. But in each one, this is a human a granuloma from a human lung tissue, granuloma from a rhesus macaque, and granuloma from an MTB-infected mice. And irrespective of the colors, the T cells are segregated from the region of MTB-infected cells. So what we then know is CD4 T cells need to make direct contact with an infected cell, but the CD4 T cells and infected cells are segregated from one another. So we were interested then in what are the mechanisms that make that spatial segregation happen, and what accounts for the failure of CD4 T cells to interact with MTB-infected cells. So we generated at least 7 different hypothetical mechanisms. One, they may just not be attracted to the region by a chemokine gradient. There might be a chemokine gradient or chemokine expression that attracts cells or distracts cells into the wrong regions. Following the precedent for neural guidance molecules that also apply in immune responses, T cells could be actually repelled from the granuloma core. There could be a negative signal that drives them away. The granuloma core environment could cause the death of T cells. In other words, they seem to be depleted or not able to migrate in, but actually they do migrate in, but then they die. So they appear to be excluded. Another possibility is a physical barrier. So many of you were probably taught many years ago that granulomas wall off the infection and cause the bacteria to be localized to a granuloma. Well, that wall may also then serve as a barrier to T cell entry. There could be insufficient antigen presentation by infected cells. So an interesting study some years ago showed that in a chemokine gradient where T cells are migrating in a gradient, if they encounter an antigen-presenting cell that carries their cognate antigen, they'll stop. In other words, a T cell receptor mediated signal will override the chemokine signal and cause basically an arrest of cell migration. So if the antigen presentation is insufficient, that stop signal might be missing. And finally, I won't go into this, but we had shown around the same time that MTB antigens can spread to uninfected cells that arrest antigen-specific T cells outside the granuloma core. So we found that MTB-infected cells export MTB antigens for presentation by uninfected cells. So with respect to the ectopic chemokine gradient hypothesis, Zach Howard generated some evidence that actually is in support of the hypothesis. So CXCL9 and its very closely related CXCL10 chemokine are chemokines that interact with the chemokine receptor CXCR3 on effector T cells and serve as a major stimulus for migration of effector T cells to tissues. So what Zach found is that the CXCL9, for some reason, the pad here goes in the opposite direction, makes the goes, there we go, whoop, and then that happens. So what Zach found is that CXCL9 and CD4 T cells co-localize in the periphery of TB granulomas. This is in an SP140 knockout mouse where things are kind of a little bit better circumscribed. So if there's no other stimulus in that core, then there would be evidence that this chemokine, ectopic chemokine expression hypothesis might be operative. On the other hand, we don't know what came first. In other words, CXCL9, CXCL10 are induced by interferon gamma, which is a product of T cells. So it's also possible that T cells are there for another reason, make interferon gamma, induce the chemokine, and the chemokine isn't what determined their localization. So the solution to this then became apparent gradually in terms of real-time intravital microscopy, which was being adopted throughout basic immunology studies to study T cell priming, to study T cell trafficking, and real-time visualization of cell migration and interactions generated using intravital microscopy has become the stuff of textbooks. I mean, we've all been taught about leukocyte rolling, arrest, and migration to the tissues, the cell adhesion proteins, the chemokines and chemokine receptors that mediate that event. Likewise, the dynamics of antigen-presenting cell T cell interactions have been understood in a far better mechanistic way, thanks to being able to really visualize the events in live tissue, in tissues of live mice. And furthermore, more recently, the mechanisms that limit T cell immunity and T cell efficacy in tumors have been pretty well elucidated. Now, in TB, there are at least two problems, two challenges that have meant that we really haven't been able to move this idea faster than we could. The first is that when you're imaging a lymph node in a live mouse, it's not too hard to hold that lymph node still, because lymph nodes don't ordinarily mouse. As long as the mouse doesn't move, the lymph node wouldn't move. But in a live mouse, the lungs are moving. And so that presents a very substantial challenge, because what you're trying to visualize are cell migration and cell interactions in a tissue that is moving. And mice breathe faster than we do. And so this is not a trivial problem. Now, just to make things even more interesting, you have to, for TB, of course, you have to do this in a BSL-3 facility. So it took about 5 years of kicking around this idea and thinking, well, wouldn't it be great to have a multiphoton live intravital imaging system in a BSL-3 facility? It seems like a long distance dream. But then I learned that Olivier Nérol and his group at the Institute for Pharmacology and Structural Biology in Toulouse had built a custom-made intravital multiphoton intravital imaging system in their BSL-3 facility. I'm not going to walk through all of these features, just to say this was not a simple task. And so Olivier and I then met at a meeting, I think it was in late 2017, just before my move here, and discussed how we could share our resources, because we had generated fluorescent protein expressing bacteria. We had generated reporter, so mice whose T cells, with a reporter that reports on T cell activation. And this would combine nicely with the ability to visualize T cell activity and interactions in BSL-3 or in multi-photon imaging. And so now it became actually a practical reality. We wrote an R21. We got the R21 grant funded, and a week after the funding began, the pandemic shut down the facilities in France for about 6 months. This is a familiar story to all of you because you all have your own versions of it. But suffice it to say that things got reconstituted. So here's the basic experimental setup. So the mice in the videos that I'll show you samples of are mice that have a genetic construct so that their background cells are red, but because of use of a macrophage-specific Cre recombinase, macrophages have green membranes. So as you look at the videos, one consistent feature of each of the videos is that the macrophages are green. Into those, then we put M turquoise expressing M tuberculosis. It actually took us a fair bit of time to find the right spectral combination of fluorescent proteins that was consistent with the multiphoton imaging system and consistent with the other fluorophores that we had for host molecules. Then we added T cell receptor transgenic CD4 T cells specific for MTB infected and an MTB infected antigen that we had generated here. So this was a large matter of sharing resources and sharing facilities to get this project going. So what I'll show you is 4 videos, and each of them has a long introduction like this, longer than I could trim away. So in this first one, keep your eye on the green macrophages, the red CD4 T cells. The vessels are gray, that's pseudo-colored. And here we go. So again, there's a long lag for each one of these, and for some reason I wasn't able to trim these long lags out beforehand. But it'll pick up here in a few seconds. So the red are T cells. This one is in a blood vessel and moving fairly rapidly. The extracellular T cells can be either mobile or sessile or arrested. This is an example of first the mobile ones and then arrested ones. We'll get back to one of the features there. And so this can be quantitated. And what we found is that after adoptive transfer of Th1 polarized CD4 T cells, at the time of imaging, which is usually about 3 days later, about 90% of the cells are extravascular, in other words, in the lung parenchyma, and about 10% or so are in the lung vessels. Of the extravascular or parenchymal T cells, about 60% are arrested and about 40% are still mobile, migrating around. So this next video shows interactions between T cells and infected macrophages. And if I can get it to snow, okay. If I can get it to play. Now that. Okay. So again, the macrophages are green, the T cells are red. And what you see is that they're basically, the T cells are moving around. So they're not dead or die. So that is one of the hypothetical mechanisms that we could reject. And some of the T cells actually arrest on MTB-infected macrophages. The MTB here is purple, and the SHG, by the way, is a sort of free signal. So that stands for second harmonic generation. That represents collagen fibers. And because of the fibrillar nature of collagen fibers, they actually will emit long wavelength fluorescence. And so basically, without being able to stain or use as fluorescent probe for collagen, you kind of get a collagen signal for free in multiphoton imaging. And so that'll come up in a bit. So of the cells that interact with MTB-infected macrophages, or at least the cells that are in the neighborhood of infected macrophages, fewer than 20% actually make contact with them. In other words, they migrate around and they just don't seem to recognize that there is a cell there that they might be interested in recognizing. So they're sort of missing an opportunity in certain cases. So finally, or not quite finally, but I'll show you example of some of the behaviors of the T cells with uninfected macrophages, and show you that many of them are motile, kind of seeking a reason to stop. The purple streaks there are collagen fibers. The purple dots that are inside green cells are the mycobacteria. In this case, they share a color in the movies. And so what this indicates is that there are some cells, some T cells that arrest on MTB-infected macrophages. But in fact, it turns out to be not a common event, consistent with the still photographs. But at least it tells us that they're sort of there, they're viable, they're able to migrate, they're not fixed in space. And so most of them are actually arrested. Now, the interesting feature then comes up with a control experiment. So I told you that we had T cell receptor transgenic mice that are specific for MTB. So the question was, how many of these overall behaviors are antigen specific? In other words, how many of them relate to the fact that the T cells that we adoptively transferred are specific for an MTB antigen? So we did similar parallel experiments with OT2 cells. OT2 cells are TCR transgenic cells specific for ovalbumin. And we know from background experiments that we've done over the years, those are a nice, clean, silent control because mice don't express chicken ovalbumin, and mycobacteria don't express chicken ovalbumin unless you actually try to make them do it. But the surprise was that the frequency of arrest of CD4 T cells was the same regardless of the CD4 T cell antigen specificity. And so what we're seeing with respect to overall T cell behavior is T cell behaviors that are responses that are irrespective of the T cell antigen receptor. And so more likely driven by things that cause T cells to migrate, that cause T cells to arrest, other than recognizing an infected cell. So finally, we got an interesting clue from that free signal from the collagen. And in this case, the macrophages are a little bit different color. The collagen fibers now are green. This is mislabeled. The collagen fibers are green and the T cells are purple. And so what you see are two phenomena. One is T cells that are migrating along collagen fibers. And in addition, you see examples of T cells that are actually arrested on collagen fibers. So T cells, particularly effector T cells, have collagen receptors. CD44 is one of them. Alpha-4 beta-1 integrin is another prominently expressed one. And those would be expressed on effector T cells regardless of the antigen specificity. And so one working hypothesis now is that collagen, which we know from other studies, that there is extracellular matrix remodeling in TB granulomas, regardless of the species. One possibility is increased collagen deposition that then basically kind of takes over as a guidance molecule for effector T cells that are recruited into the lungs. Now, to make things even more interesting, Dan Barber, who is at the intramural program of the NIAID, recently did single-cell RNA sequencing of T cells isolated from granulomas from MTB-infected non-human primates, and made, to me, the pretty surprising discovery that certain effector T cells actually make collagen, which I don't think the textbooks mention T cells making collagen, but he now, I think, has pretty persuasive evidence of that. And so here you see that certain of the T cells, this is just replaying again, certain of the T cells associated with collagen fibers are moving and migrating along the collagen fiber. Others are arrested on the collagen fibers. So it may very well be that collagen signals are major determinants of the migration and the localization of MTB infected cells and their relationship to CD4 T cells. So from this, I think we have excluded at least a couple of those possible mechanisms. We don't see any evidence that T cells are excluded from the granuloma core. They just don't hang around there. In other words, they don't dwell there long, so they look like they're depleted, but they're migrating through and they're transient. They don't die. We haven't seen any evidence of that. In terms of the insufficient chemokine gradient, that still remains a possibility. In other words, maybe there just isn't a good, strong signal to attract the bulk of the population of T cells to the granuloma core. The ectopic chemokine signal is one that I think now is tractable to begin to test. The physical barrier may be what the collagen deposition and extracellular matrix remodeling might represent. But then there are the cells that do get into the region and that actually do interact with MTB infected and actually MTB uninfected cells. So the next series of experiments will then involve our T cell activation reporter specific T cells to say, okay, of the arrested T cells on TB infected versus MTB uninfected cells, how many of those T cells are actually getting activated through their TCRs? And finally, I think our old model of MTB antigens being spread to uninfected cells may account for at least some of the interactions with uninfected cells by the antigen-specific T cells. So like I said, it's a work in progress. It's far from finished. There are no conclusions here. It has taken however many years it's been so far, about 15. And I mentioned that just for those of you who are younger and earlier in your careers, that, you know, sometimes it takes a while to pull things off. You know, at some point, I've got the fanciful notion of establishing this capability in a BSL-3 facility in one more location other than Toulouse. Toulouse is not a bad place to visit. Let me make very, very clear. But it's a long distance. We have weekly calls that I think are really very productive, and it's kind of helped things move along. I'll tell one other anecdote, and that is early on when Olivier and I were discussing this, getting started, there was another individual from his institute, Antonio, who had designed the multi-photon imaging system in the BSL-3 facility and really was the only one who knew how to operate it. And in a call one day, Olivier said, Antonio is leaving the institute. Well, you know, this is very bad news. But Olivier is not a physician. Like a good physician, he delivered the bad diagnosis and then gave me hope. He said, we're working on recruiting somebody that I think will be able to take over the project. I said, okay, you know, I trust you. Well, it turns out the person he recruited was the person who developed the ability to do multiphoton ultravital imaging on lungs when she was a postdoc with Max Krummel and Mark Looney at UCSF. And so Emma Lefrançais is now the person who has taken over the project and is I would say, more than capable. She was the best person in the world to recruit to take it over. So sometimes there are bumps in the road, and sometimes you bounce higher than you were. So that's all I have as today's guest speaker. Thank you. Happy to take questions, suggestions. Sarah. Oh. My question for Val is, where can we get those T-shirts?
Great talk, Joel. Did the OT2 cells also hang out around the collagen, or was that specific to the OT2 cells? Did they also hang out with the collagen, or just
so the question was about OT2 cells.
Just as a control, yeah.
They behaved similarly, but they were similarly primed in vitro, and so we're not surprised that they and we wouldn't expect antigen specificity to have much to do with adhesion protein.
Maybe the functional properties might be.
I'll also mention one other unpublished, but I think about to be published result that is, I think, could be relevant here. Rasmus Mortensen at the Staten Serum Institute has done what a lot of, he's done things in a little bit different way. Some groups studying MTB infection and T cells will gate on CD44 positive cells and think that they're the only ones that count. Rasmus does not gate them out and has discovered that actually CD44 negative T cells enter the lungs of MTB-infected mice and contribute to protection. Now, CD44 is one of the collagen receptors, and so it's possible that people are gaining out interesting cells. So we are also kind of now incorporating Rasmus into the sort of experimental team.
Thank you for a great talk. Joe, so this TB granuloma simply reminded me like a tumor immunology, right? So is it like this TB granuloma causing a high PD-L1 expressing antigen presenting cell, also like a causing hypoxia, hypoxia like a tumor, low oxygen antigen makes in a T cell resting state, right? So someone tried the immunocheckpoint block.
I understood your question correctly.
No, you're hearing good.
But with respect to co-receptors activation,
or something, yeah, immunocheckpoint, yeah.
We know that in TB, mice, non-human primates, also people, PD-1 blockade is actually mechanism.
Oh.
Now, you would like to think one mechanism of limited TB suscitate them on cancer tumors. Dan Barber was certainly focused. But it turns out that PD-1 is an essential negative regulator. And in the absence of PD-1 or the presence of PD-1 blockade, basically there is out of control CD4 T cell mediated infection. And so TB is more complex.
Yeah, it seems complex. But also collagen is very interesting thing. So one of my Japanese friends, he's researching HIV, but actually CD44, antagonist collagen is capturing HIV virion, and the CD4 get there, right? So it's anti-CD44 making physically contacting the HIV and the CD4 T cell, then now acting with the TB, right? So it's a very making a complex of a TB HIV co-infection. Yeah. Yeah.
Hi, Joel. Very awesome work. I guess also related to Yoshi's point of doing a deep dive in like cancer literature and how they're trying to identify like the in vivo signals that differentiate monocytes into macrophages and tumors are immunosuppressive, I guess. For your possible mechanism, do you think that something similar is happening for the recruited macrophages? Monocytes are differentiated more into macrophages, and maybe there's not enough DC differentiation to like reactivate the T cells in the lung, and that's why they're not.
So that is the topic of quite a bit of other work in the lab, because we really want to understand what is the potential of T cells if we can get them to the right place. Get them to see the right things. We don't, another limitation in the field, we don't know the best antigens for T cells to recognize. And so we might be studying something that is true for T cells, specific for secreting into the antigens, but maybe not generalizable. Those are the only two TCR transgenes we have. But for certainly, as you know, not every antigen from MTB has T cells with some properties.
Do we have more questions?
Okay.